Hello viewers, welcome to the course Elements of Genetics, and in today's part we shall be learning about cytoplasmic inheritance and the interaction of plasma and nuclear genes. The Mendelian inheritance pattern involves genes that directly influence the outcome of an organism traits, and they obey Mendel's laws. Most genes in eukaryotic species follow a Mendelian pattern of inheritance. However, there are many that do not. Linkage can be considered as non-Mendelian inheritance. Patterns of inheritance that deviate from a Mendelian pattern, they are due to maternal effect and epigenetic inheritance, or there can be extranuclear inheritance. In case of maternal effect, they involve genes in the nucleus. In extranuclear inheritance, they involve genes in organelles other than the nucleus, that is mitochondria and chloroplast. Let us appreciate one point here, that in the entire sequence of all the parts of this course of Elements of Genetics, the reference point is of Mendelian genetics, that whether all the principles we try to learn, they follow Mendel's law or they do not follow Mendel's law. Those exceptions which came out that they do not follow the trend shown by the Mendel, they also refer to that principle and confirmed indirectly that Mendel was right in the sense that the genes were located separately on separate chromosomes and they segregated and assorted independently during the process of the germ cell formation. Here in this part, we are exposed to yet another kind of the exception to the Mendel law, where the maternal cytoplasm or maternal influence overrides other effects of the chromosome or the nuclear genes. Maternal effect means that it refers to an inheritance pattern for certain nuclear gene in which the genotype of the mother directly determines the phenotype of her offspring. This phenomena is due to the accumulation of gene products that the mother provides to her developing egg. The point to understand is that after the fertilization, the egg or zygote, that is the product of fertilization, develops in the mother's body, and therefore, there is a contribution of cytoplasm from the mother, whereas the nuclear gene is donated from the father. The first example of maternal effect gene was discovered in 1920 by A. E. Boycott. He was studying morphological features of the water snail, the Limni peregra. In this species, the shell and internal organs can be arranged in one of two directions, that is, either it is right-handed or it is left-handed. The right-handed is referred as dextral and left-handed is referred as sinistral. The dextral orientation is more common and dominant. The snail body plan curvature depend on the cleavage pattern of the egg immediately after fertilization, which is shown here in the reciprocal cross, that we can see the dextral pattern and the sinistral pattern. The dextral pattern is referred by the capital letter, means the dominant gene, and in F1 generation and F2 generation, and after the result of the crosses, what we observe that there are a ratio of Mendelian ratio, that is 3:1. A phenotypic ratio would be predicted by a Mendelian pattern of inheritance. Alfred Sturtevant later explained the incongruity with Mendel inheritance, that snail coiling is due to a maternal effect gene that exists as dextral capital D and sinistral small d alleles. These phenotypes of the offspring depended solely on the genotype of the mother. His conclusions were drawn from the inheritance pattern of the F2 and F3 generations. Now, it ultimately became clear that this structural orientation of the snail body, that is in terms of dextral or sinistral or right or left orientation, they primarily depended upon the structure which was present in case of the mother parent, which is shown here in this diagram, and a analysis of F2 and F3 generation. The analysis of this diagram would ultimately show that whatsoever the structural configuration or the genetic constitution, the structural orientation, that is left and right position of the snail, would primarily be determined by the structure of the mother parent, and this situation has led to the identification and the coinage of the term cytoplasmic inheritance, which is contributed from the mother primarily. These again are the diagrams showing the same thing, that the fact that the contribution in the orientation of the shape of the snail is determined by the maternal parent. Now, it is to be noted that the phenotype of each generation depends on the maternal genotype of the previous generation. The capital D or capital D small d combination mothers produce dextral offspring, the small d mothers produce sinistral offspring. The phenotype of the progeny is determined by the mother's genotype, not the phenotype. The genotypes of the father and offspring do not affect the phenotype of the offspring. These are the conclusions of the snail's experiment, and they point out toward only one thing, that finally the shape is determined by the genotype of the mother, which is primarily the cytoplasmic contribution or the genes present in the cytoplasm. The gene products are reflection of the genotype of the mother. They are transported to the cytoplasm of the oocyte, where they persist for a significant time after the egg has been fertilized, thus influencing the early developmental stages of the embryo. This is a figure which clearly shows that the nurse cell from the cytoplasm, they encircle or surround the oocyte, the female egg, and it changes its genetic constitution. The capital D gene products cause egg cleavage that promotes a right-handed body plan, and all spring, in case of mother is a capital DD, all springs are dextral, and when mother is a capital D small d, all springs are still dextral, and the small d gene products cause egg cleavage that promotes a left-handed body plan, and even if the egg is fertilized by sperm carrying the capital D allele, the sperm genotype is irrelevant, because the expression of the sperm gene would not be too late. So, here there is a clear-cut influence of the maternal cytoplasmic inheritance can be visualized here in this experiment and the diagrams. The epigenetic inheritance refers to a pattern in which a modification occurs to a nuclear gene or chromosome that alters gene expression. However, the expression is not permanently changed over the course of many generations. The epigenetic changes are caused by DNA and the chromosomal modifications. These can occur during oogenesis, spermatogenesis, or early embryonic development. The extranuclear inheritance refers to inheritance pattern involving genetic material outside the nucleus. The two most important examples are mitochondria and chloroplast. These organelles are found in the cytoplasm. The extranuclear inheritance is equal to cytoplasmic inheritance. The deep probe and further studies in case of the maternal influence of the genes or the cytoplasmic inheritance has led us to a point that the extranuclear inheritance is synonymous to cytoplasmic inheritance. In general, mitochondrial genomes are fairly small in animals. They are intermediate in size in fungi, algae, and protists, and they are fairly large in plants. This is a diagram of the mitochondria where the DNA is present inside the matrix of the mitochondria, and in the left side of the mitochondria the DNA, which is circular in nature, is shown here, and this DNA which is present in the mitochondria is responsible for the maternal influence, maternal inheritance or the cytoplasmic inheritance. The main function of mitochondria is oxidative phosphorylation, as we know, that is respiration, and a process used to generate ATP or adenosine triphosphate. The genetic material in mitochondria is referred to as empty DNA or mitochondrial DNA. The human mitochondrial DNA consists of only 17,000 base pairs. It carries relatively few genes, that is ribosomal RNA and transfer RNA genes, 13 genes that function in oxidative phosphorylation, and the most mitochondrial proteins are encoded by genes in the nucleus, and these proteins are made in the cytoplasm, then transported into the mitochondria. This is the example of the interaction between nuclear genes and the extranuclear genes. The extranuclear genes which are present in the organelles like mitochondria and the chloroplast, but some of the genes are synthesized in the nuclei and responsible for the function of the respiration, whereas some of the genes which are synthesized in the mitochondria, they also control the activities of the nuclear genes. So, these are the examples of the interaction. And the main function of chloroplast is photosynthesis. The genetic material in chloroplast is referred to as cpDNA or chloroplast DNA. It is typically about 10 times larger than the mitochondrial genome of the animal cell. The cpDNA of tobacco plant consists of 156,000 base pairs. It carries between 110 and 120 different genes, ribosomal RNA and transfer RNA genes, and many genes that are required for photosynthesis. As with mitochondria, many chloroplast proteins are encoded by genes in the nucleus, and these proteins contain chloroplast targeting signals that direct them from cytoplasm into the chloroplast. Similar to the mitochondria, the behavior of chloroplast is like that and responsible for maternal influence or cytoplasmic influence, as well as example of the interaction between nuclear and cytoplasmic genes. The advancement in the understanding of cytoplasmic inheritance was further contributed by research on an ornamental plant that is known as Mirabilis jalapa, the 4 o'clock plant, and Carl Correns discovered that pigmentation in Mirabilis jalapa shows a non-Mendelian pattern of inheritance. Leaves could be green, white or variegated, with both green and white sectors. Correns determined that the pigmentation of the offspring depended solely on the maternal parent and not at all on the paternal parent. This is termed as again maternal inheritance or the cytoplasmic inheritance. This is the diagrammatic representation of the various type of crossing, and we can see that whatever is the color in the female parent is the color in the offspring. So, if it is white, the offspring would be white, if it is green, the offspring would be green, and if it is variegated, the offspring would be variegated. So, there is a contribution of the leaf coloration from the maternal parent only. In this example, maternal inheritance occurs because these chloroplasts are transmitted only through the cytoplasm of the egg. The pollen grains do not transmit chloroplasts to the offspring. The phenotype of leaves can be explained by the types of chloroplasts found in the leaf cells. Either they are green phenotype as wild type, white phenotype is the mutant, and a cell can contain both type of chloroplasts, which leads to the heteroplasmy or the variegated type of the leaf structure. In the context of cellular explanation for the variegated phenotype in Mirabilis jalapa, that we have to consider a fertilized egg that inherited two types of chloroplasts, that is green and white. As the plant grows, the chloroplasts are irregularly distributed to daughter cells. Sometimes a cell may receive only white chloroplast. Such a cell will continue to divide and produce a white sector. Cells that contain only green chloroplast or a combination of green and white will ultimately produce green sectors. This is a diagrammatic representation of the variegated leaf, and the different portions shown here, that white portion will only contain white chloroplast, that is leucoplast, green with green chloroplast, and the inheritance pattern of the leaf will ultimately depend upon the type of the leaves which has present on the maternal plants. Another proof of the cytoplasmic inheritance with experiment with yeast, and this is known as the Pittite trait in the yeast. The Pittite means the slim or the slow growing or hungry yeast. Mutations that yield defective mitochondria are expected to make cells grow much more slowly. Boris F. Russi and his colleagues identified Saccharomyces cerevisiae, that is yeast mutants, that have such a phenotype. These were called Pittite mutants because they formed small colonies, and wild type strains formed larger colonies. This is shown here in the diagram. The wild type has the larger colonies and the mutant type has small colonies which are known as Pittite colonies. The yeast come in two mating type A and alpha, and thus Euphrasia was able to cross yeast belonging to two different strains, and the diagram shows the distribution of the wild types and the Pittite types. The progeny have both wild type and the neutral Pittite, and the progeny have both wild type and suppressive Pittite mitochondria. So, how come only Pittite colonies are produced? There are two possibilities, and out of these two possibilities the conclusion has been drawn. The inheritance of the Pittite gene has been contributed from the maternal parents and not from the male parents, which has been referred in case of the yeast. Now, the inheritance of chloroplast in algae, the unicellular alga Chlamydomonas reinhardti is a model organism to study the genetics in case of unicellular alga. It contains a single chloroplast. It occupies more than 40% of the cell volume. The most strains are sensitive to the antibiotic streptomycin, which is referred as SM superscript S, and in 1954 Ruth Sager identified a mutant that was resistant to streptomycin, which is referred as SM superscript R. Like yeast, Chlamydomonas can be found in two mating types. Mating type is due to nuclear inheritance. Sager and colleagues discovered that resistance to streptomycin was not inherited in a Mendelian manner, and in subsequent studies they mapped several genes including the SMR gene to the chloroplast chromosome, and that is why this diagram again shows the distribution of the resistant type and susceptible type, and the conclusion has been drawn that again this is on account of the contribution of cytoplasmic inheritance or the genes present in the chloroplast of the Chlamydomonas. The pattern of inheritance of organelles. The pattern of inheritance of mitochondria and chloroplast varies among different species. They are heterogamous species, they produce two kinds of gametes, and in these species organelles are typically inherited from the mother. The pattern of inheritance includes that species with maternal inheritance may on occasion exhibit paternal leakage. The paternal parent provides mitochondria through the sperm. In the mouse, for example, 1 to 4 paternal mitochondria are inherited for every 100,000 maternal mitochondria per generation of offspring. But this is a very rare phenomena, and primarily the cytoplasmic inheritance is synonymous to the maternal inheritance. One of the offshoot of this cytoplasmic inheritance is in the form of endosymbiosis theory, which is related to the origin of life. The endosymbiosis theory describes the evolutionary origin of mitochondria and chloroplast. These organelles originated when bacteria took up residence within a primordial eukaryotic cell. The endosymbiotic origin of organelle is supported by several observations. These include organelles have circular chromosomes like bacteria, and organelle genes are more similar to bacterial genes than to those found within the nucleus. In fact, the endosymbiosis theory suggests that when primordial cell started to develop in an aquatic situation, there were certain prokaryotic organisms or bacteria-like organisms which started to reside in that eukaryotically developing cell, and these bacteria, if they were non-photosynthetic, ultimately became mitochondria, and if they were photosynthetic, they became chloroplasts. So, this is the theory. Endosymbiosis means the organisms residing within the kind of cell and developing their itself and developing into different kind of organelles. The presence of DNA in mitochondria and in chloroplasts is the evidence of this endosymbiosis theory. In today's part, we try to learn about the cytoplasmic inheritance, that is particularly the influence of the maternal cytoplasm. In certain experiments, scientists and geneticists observed that the genetic pattern or the segregation pattern in F2 and F3 generation were not as per the Mendelian pattern. This was particularly observed by Bicot in case of the structural orientation of the snail body, that is dextral pattern or the sinistral pattern or the left pattern or the right pattern. And the analysis ultimately led to a particular observation that the body pattern mainly depended upon the genotype of the maternal parent and not of the paternal or from the father. So, this kind of influence was referred as the maternal influence, and the further study showed that the cytoplasm of the maternal parent influenced the germ cell and changed their genetic constitution. The cytoplasmic factors from the mother's parent, they influenced the germ cells and changed their constitution, so that ultimately the phenotypic expression of the offspring depended on the genotype of maternal parent. Further researches on the plant system, particularly the Mirabilis jalapa and the algal system like Chlamydomonas and the yeast system, the Cerevisiae, they all refer to certain kind of pattern in case of petite colony of the yeast, as well as in the type of the chloroplast of the Chlamydomonas, as well as the variegated leaf structure, all showed one common thing, that the genotype would adopt the structural pattern of the maternal parent. And the reason was found out by the fact that certain extranuclear genes were present in case of chloroplast and mitochondria, and they contributed toward the expression of the phenotype in the offspring, and particularly they adopted a pattern which was present in the mother. So, the maternal inheritance became synonymous to extranuclear inheritance as well as cytoplasmic inheritance. The offshoot was observed in case of the endosymbiosis theory, that when eukaryotic cell was developing and evolving, there were certain bacteria, the photosynthetic bacteria and non-photosynthetic bacteria, which started to reside in that developing eukaryotic cell and became chloroplast and mitochondria, depending upon the fact that whether they were photosynthetic or non-photosynthetic. So, I hope at the end that you might have enjoyed this lecture.